Dear student, welcome to the course of Basic Genetics. Myself Dr. Richa Arya, and we are talking about the gene mapping on eukaryotic chromosome in this unit, and this is our last lecture where we are going to talk about how we can map the genes in yeast and Neurospora by using tetrad analysis. In this class we are going to discuss about gene mapping in yeast and Neurospora using tetrad analysis. These two organisms that we are going to discuss in today's class, they are a bit different than the ones that we have discussed previously. Most of the organisms we have discussed in previous class, taking the example of Drosophila or maize or other examples that you might have been doing for your practice, they are mostly diploid 2N. These fungus are actually following the life where they have a haploid phase as well as diploid phase, right. So, we are taking advantage of their haploid phase to understand the order of genes on the chromosomes. So, the tetrad analysis in certain haploid organism are being done by counting the spores in the ascus. So, ascus is a structure where the spores are encapsulated inside a covering, right. So, this structure is called ascus. We will show you when we cover the life cycle, we will show you what exactly it is, and these spores are haploid. So, if we look at the life cycle of yeast, so we are starting with yeast, and I want you to pay attention to the unordered spores, right. So, we will be taking the two examples, one for yeast, another for Neurospora, and yeast has unordered spores whereas Neurospora has ordered. What is the difference? Do not worry about it. I will make it sure that you understand the difference very clearly, but just remember these 2 points, that in yeast they are unordered and in Neurospora they are ordered. So, what does the yeast life cycle look like? If you see, then you will find that, as I said before, that they have haploid and diploid life cycle. It is very clear actually in case of yeast. You can see that when these two different mating type A and alpha, they fertilize and they make a 2N zygote, and that what this 2N can do, it can divide and vegetatively propagate as 2N, right. And this 2N organism, what it can also do, it can also form spores by undergoing meiosis. So, when it undergoes meiosis, then it forms 4 spores. We know that one meiosis produces 4 cells. So, when it undergoes meiosis, it forms 4 spores, and these 4 spores are covered or encapsulated, and this encapsulated structure with the spores is called as ascus. So, we are going to analyze this ascus, the ascus of 4 spores, right. Both are all the spores are haploid. So, the 3 ascus types that we find in the yeast when we are having 2 genes in question. So, you see we have this AB when it is crossed with A plus B plus, what combination type we are expecting. You see, just do not read these names now, I will explain you what these names are, just focus here. So, if you see that there are AB parental type, AB parental type, A plus B plus parental type, A plus B plus parental type. So, they all are the parental combination. That is why this is called parental ditype, PD, parental ditype. So, this is the number. How many parental ditype ascus were seen? And then come to the non-parental ditype. So, non-parental ditype, why they are being called non-parental? Because see, A, B plus, none of them has this combination. A, B plus same, not present. A plus B, not present. A plus B not present, right. So, none of the combination is matching with the parent, that is why non-parental ditype. Then comes to this class, what you are seeing? Here you are seeing a parental type. This one is also a parental type, one is this, another is that, and these two are recombinants. So, these two, they are recombinant type. So, because this kind of ascus has 4 spores where you see parent as well as recombinant type, it is called as tetra type, TT. So, if we want to calculate the map distance between this A and B gene, what we will do in case of yeast? What we will do is, to calculate the map distance, we will half the number of tetra types, right. So, because you see, just to compensate for the non-recombinant that are present inside, we are reducing this number by half and adding the total number of non-parental types. So, parental type combination we do not need, right, because they are non-recombinants. Whenever we are checking the distance between the two gene, what we care for? The total number of progeny and the recombinants, and then we divide it by total number of asci and then multiply with 100. So, for in this case, what we see is that map unit is 1 by 2, 20 plus 5, that comes to 15. So, this is the distance between the two genes, right. That is how we calculate the map distance. Now, come to the understanding about the parental ditype and non-parental ditype. So, see, parental ditype, again, when parental ditype are more compared to the non-parental. Non-parentals are what? Non-parentals are recombinants. In non-parental class, we see the recombinants. If we do not have the recombinants, then it clearly indicates that the genes are highly linked, right. So, that is what I have written here on this slide, that when parental ditypes are more than non-parental ditype, then genes are on homologous chromosomes because of the rarity of non-parental ditype, which actually arise from the 4-strand double crossovers. The tetrad types arise from single crossover. Why we are saying it is arising? Because 2 of them are parental type. So, which means that these strands, one chromosome have 2 sister chromatids. So, one of them actually never undergo recombination. I will show you in a minute. So, if the genes are on non-homologous chromosomes or if they are greatly apart, same thing again on the chromosome, then you will start seeing parental type equal to non-parental type because of the independent assortment or crossover in various combinations of the chromatids. So, now just so that to help you understand this concept, how exactly these kind of combination are arising due to crossing over, how beautifully you can see in haploid organism. So, what is the advantage of haploid organism? See, before we always have to set up a test cross, we have to always bring our combination with a recessive set of genes so that we can start seeing what is there in our parental combination, right. But here, if the organism is haploid only, then one copy of gene has enough capability to express itself and you do not need to make the experiment more complicated. So, what you are seeing here, that these 2 homologous chromosomes carrying the gene A and B and then A plus B plus, if there is no recombination, all the spores you are getting are parental type. You see, if there is one recombination, now pay attention, it is very easy. If there is one recombination, then you have this parental type sister chromatids that eventually become chromosome without any recombination, and you have 2 recombinant, and that is why you have tetra type. If you have 2 recombinations, if you are 2 crossing over but involving 2 sister chromatids, then what is happening? It is also creating only parental ditype because one recombination is bringing the change and another one is going to bring it back, right. So, that is why only a small piece got exchanged, and if you do not know what genes are there, then you have no way to really recognize if anything had happened or not happened there. And that is why you will have all the parental type combination for the genes that you are interested in studying. If again there are two crossover but involving 3 sister chromatids, see there are 3 sister chromatids involved in this recombination. And when 3 recombinations sister chromatids are involved, you get the Tetrad type. So there are one combination here, another here, and that is when you start getting the Tetrad type combination. Another, you see, this is also 2 crossing over. In the previous example, you can see one sister chromatid was undergoing crossing over with both the sister chromatids of the other homologous chromosome, and here one arm with one sister chromatid and another with the same sister chromatid of another homolog. So in this case also you are going to get the Tetrad type. So they are just alternate conditions of the crossing over. And the fourth condition is when you have the involvement of all the 4 sister chromatids, right. So this one is recombining with this and these 2 are together, and in that case remember I told you that non-parental ditypes are formed when all the 4 chromatids are getting involved in crossing over, and that is when you have no parental type, all recombinant type in this class. So linkage is indicated by an axis of parental ditype over non-parental ditype. It is just a summary so that you remember what we learned so far. And if we solve a problem, just to revise and revisit what we learned by using this question, where in a yeast the histidine locus is a gene for an enzyme in the synthesis pathway for the amino acid histidine, and the lysine 11 locus is a gene for an enzyme in the synthesis pathway for the amino acid lysine. A haploid wild type strain His5 plus and Lys11 plus is crossed with the double mutant His minus, His5 minus and Lys11 minus. The diploid is allowed to undergo meiosis, and 100 ASCII are scored with the following results. So you already have the results for different type of ASCII. The first thing that you need to know what type they are, like Tetrad type, right, parental ditype or non-parental ditype. This is important for you to identify. So that only you will be able to understand the linkage arrangement. So if you see, you can clearly see that this is a parental ditype. Why? Because these 2 are exactly the combination present in the parents. And this is non-parental ditype. See, there is none, all are recombinants here. No one is parental type combination. And these one are Tetrad type because you have a parental type here, you have a parental type here, and these 2 are recombinant type. Right. So linkage arrangement of these loci, what is the linkage arrangement of these loci? You need to calculate the, apply the formula by, you remember I told you the 1 by 2 and then Tetrad type plus NPD, finally divided by total progeny and then multiplied by 100. Right. So you can calculate it this way. So now come to the next fungi that we need to discuss for the ordered spores, which is Neurospora. So in case of Neurospora, it is also running a haploid lifestyle by and large, right. It is also produced 2 mating types, and we are taking example of A and alpha and then A and A small genes that we will be discussing in the combination. When the 2 spores they fertilize, then they make 2N ascus. When 2N ascus undergo meiosis, then they become N, and when they finally divide, you have a ascus with 4 spores. But what is different compared to yeast is that the Neurospora go one more round of mitosis, and so the 4 spores turn into 8. So here you have 4 spores and here you have 8, right, and I will show you in detail, but they are all in pair of 2. So they are in pairs. So if you see how the meiosis is taking place in case of Neurospora. So it is very interesting because this is also one of those classic discoveries where using Neurospora spore arrangement, you can actually trace how exactly meiosis might have happened and how the sister chromatids have separated, because when the chromatids separate, that one separated set becomes one spore, right. So in Neurospora there are total 7 pair of chromosomes, and for the simplicity's sake I am showing only one set here. So you see this is one homologous chromosome, here is another, and you have 2 sister chromatid each. One is carrying a dominant A gene and another has a recessive A gene. So when these sister chromatids get separated, let us assume that there is no recombination there, then these 2 sister chromatids get separated, one goes on one side, another on another. Same is true for the small a and small a, right. And then what happens, this one undergo mitosis, one more round of mitosis, one more round of mitosis, one more round mitosis, one more round mitosis. So meiosis, towards the end of meiosis you have 4 sets, and after that with one round of mitosis you have 8. So you see all the A's are arranged on one side and all the small a's are arranged on one side. So if you open this ascus on one end and start removing the spores one by one, you really know what you are going to expect, right. So at least expected combinations you will be very sure, because every spore has a duplicated spore and you are also familiar with the homologs, right. So that is how they become the spores, and finally you can use this spore to understand the or calculate the genetic distance. So mapping a locus, how we are going to use, as I said, using the Neurospora. So just assume this in continuation to the previous example that I showed you before, where there is no recombination is taking place. You just see that these 2 sets, haploid sets, come together and they start dividing, and this is exactly what we are expecting. So now remember one thing, when no recombination is happening, you have the spores in a combination of 4:4. This is one point, right. 4:4 of what? A, right. And it could be reverse also, these spores could be on that side too. So A:A, that is also 4:4. So there are 2 possibilities of the arrangement of these spores within the ascus, and because there is no recombination here, we call them first division segregation, right. So in first division whatever segregation you get, and that is why all 4 on that one side are same and all other 4 are same. So remember this, you will see it is very interesting way of looking at the gene order and distance. Let us look at the another possibility where the recombination is taking place. So if the recombination is taking place, you can see the chiasma forming, you see this. If the chiasma is forming, then you can see the fragments are getting exchanged. So in meiosis 1, one sister chromatid is like parental type and another one has the recombination. The same is true for the other homologous chromosome. One strand is parental type, whereas the next one is having the recombination, crossing over and recombinant. So in the meiosis 2, when they separate, you start seeing a parental type, a recombinant and a parental type. And when they duplicate, you will have AA, small a, capital A or small a. So this is the arrangement of 2:2:2:2:2, right. So this is this arrangement, right. Or it could be possible that these reds, actually this could be 2:2:2 for starting with capital A, but it could be AA and then AA and then AA and then AA. That is also same 2:2:2:2:2. Or it could be like this, where this particular chromosome, the one in the center, they change their position. So like, you know, if this is the other way around, then you will have a combination of, so A, and A, right. So there are different possibilities of getting the orientation of spores in the ordered arrangement in the within the ascus, right. So this is how these spores can be arranged. So if this A is on the other side, this A is on the other side and this one is on this side, then you will get this kind of arrangement. So this we call second division segregation. So this is the actual condition where the recombination can be seen, right. And if there is a recombination there, then you can calculate the distance between the genes. So here the map distance will be half the number of SDS ASCII and divided by the total number of ASCII into 100. So we count for them, and why we do half again to compensate for the additional number of the spores which are non-recombinants. So let us take this example of 3-point test cross in Neurospora. So this is a 3-point test cross because we are mapping the gene AB in relation to centromere, right. So we have the chromosome, we are checking the position of in relation to the centromere, where is A and or where is B, any direction, but I am just hypothetically making them just to show why it is a 3-point test cross, because of we are mapping the genes in relation to the centromere. So we want you to calculate the distance between the gene from the centromere, the distance of A and distance of B, how we calculate it. So you see, from this cross they have given you the number of different ascus. So there are total ascus given to you are 7, and each ascus you already know that has 8 spores. So if you look at this first ascus, you can see they are all parental type combinations. So we are not interested in it. You can also see the number, it is too high, right. So it clearly shows that it is first division segregation, no recombination happens here. Now if you look at this example, right, and then you will see that here the A, if we say AB, then you have these, the total number of ascus that you get in this condition are only 2, and they all are what? Non-parental ditype, right. So they are NPT. Now come to ascus 3. What you see? You see this is the parental, this is the recombinant, this is parental, and this is also recombinant, right. So these are tetrad types, and what you also see is that here if I ask you if the position of A has changed or not, then you will see all A's are together. So this particular ascus is changing the position for B, you see BB, but here is B plus. So one of the chromatid has changed the position for B. So it is the SDS for B, right. So 101, this number is 101 for B. If you look at ascus 4, this has changed the position for A, see AA, but next is A plus and then AA. So that is why you have this recombinant for A. Here you look at this, A is there and then A has changed the position here, you see after A you see A plus, 4A plus and A, and for B2 here it is B and B, but next to it are 4BB. So it is 2:4, right. So this has changed the position for A as well as B. So that is why we put it here, and same way you calculate it for the ascus 6 and ascus 7, and these numbers if you just sum them up and divide by total, multiply by 100, you will know the distance for A, and for B the same thing, add up all the numbers, divide by 1000, multiply by 100 and then divide by half, right. Remember, in the formula I showed you that you have to make the total second division segregation, you need to divide it by 2. And that is why how you can get these numbers 8.4 and 13.0 centimorgan. And so you can arrange this these two genes on the, so if you make the centromere in the center or at one point, then you have this A at 8.4 distance apart and B on the other side of the centromere at 13 centimorgan apart. You can also make it like that. So, on the centromere you can make both the genes arranged on one side, A 8.3 centimeter apart and B 13 centimorgan apart. And this is just I just put this formula so that you remember how we have calculated the distance. So, this can be more precise when we have more genes to map around the centromere, and then we can have more specific positions with respect to each other. So, here is an activity question for you. In this question it is asked that you read the question where there's gene MAD14 that codes for an enzyme which is an amino acid in Saccharomyces, and it is very close to a centrosome. And what you need to do by looking at different combinations of PD and PD and Tetrad type, you need to answer if B is tightly linked with A, or if B is closely linked with A, or B on a different chromosome from the one A is on, or but far apart from the centromere. So, I think that you need to read it carefully and revise the concept, and same as I said before, if you face any problem solving such questions, do let me know. I will help you to go through the problem. And with that, I would like to thank. I hope that you will be able to follow the concepts we discussed in this class, and if you face any trouble, please do write us back, we will help you. And with that, thank you for your time.